Lots of food for thought in the discussion that's coming up soon. But first, we have Dr. Barry Hardy, who's CEO of Douglas Connect and president of the OpenTox Association, which is an international nonprofit promoting open knowledge community approaches to new methods in predictive toxicology. Welcome.
Thank you. Where's my control here for the slides? This guy, is it? This one here. Okay, well, I'd like to thank the National Academy of Sciences for inviting me here today. The theme of my talk is connecting people and data for decision-making and safety assessment, and is based on my experiences and learning over the last few years around an activity, a community, a framework called OpenTox, and various things it's inspired and interacted with. So my main three key messages I have in my talk is we need to invest in data practices and data sciences. It's important. We need better integration of data science to enable program goals, both scientific programs and industrial programs. And we need stronger relationships between end users and data generators. Now, most of the work that we do is on our own individual islands. And this is where our data is generated, and for the most part, where it resides. And as a result, we have several disconnects between the generation of data and its processing, and between its integration and its use. But solving major challenges today requires the engagement of all stakeholders and greater resources than any of us can wield individually. We need to build bridges between our islands. But to do so, we must somehow work together to plan experiments, describe data, and how to make it accessible, usable, and actionable for specific purposes, ranging from scientific research to industry consortia goals and regulatory acceptance of information from new methods. Now, in the early days of OpenTox 10 years ago, we specified the computer interfaces and open standards required to develop resources in predictive toxicology. The concept was, how do we specify the components we need for data algorithms and models to build applications that would be fair to data, providing access also to the models we build with them. We even realized that small discrepancies in language could result in very different meanings. We needed a common language, formally integrated within an ontology, to achieve semantic interoperability between resources. Only then could people and machines form good judgments across integrated data. Also, only if motivations were aligned and a minimal understood set of common rules existed could we achieve mutual goals. So the promise of an extensible and interoperable approach was first demonstrated by the BioClip's OpenTox integration, a decision-making tool for drug design purposes for the pharmaceutical industry, which combined the provision of distributed data and models from OpenTox into a workbench for drug design. When we founded OpenTox some years later as an international nonprofit association in 2015, we recognized the importance of open data, but also open knowledge, methods, tools, and resources supporting predictive toxicology, and also the broader goals of safety assessment and risk management. We knew that OpenTox would need to take on its own momentum. So we've been encouraging the grassroots formation now of local chapters, and the first one was just founded last year in North Carolina, and others are starting this spring. Community activities such as data hackathons and workshops where we bring data scientists and scientists together to work on data and software and tools are a tremendous, I think, way of driving our activity and advances forward. Now, ToxBank was our first OpenTox infrastructure project, and it supported SURIT, which was a public-private partnership between the European Commission and the Cosmetics Europe, representing the cosmetics industry, to advance new methods for the evaluation of repeated dose toxicity. ToxBank tackled numerous issues, such as the need to integrate infrastructure and processes, the selection of common reference compounds and biologicals used by all the partners and projects across the program, case study-driven research, and documentation and storage of all protocols used on the program linked to the data sets generated. In ToxBank, we also introduced the use of data templates, which described the experimental biological metadata based on the emerging ISA standard. We discovered, however, there were significant gaps between lab practices and the practices needed to achieve this data integration. Motivation for change by academics was varied, but even when present, was often not transferred easily into action. Barriers to data sharing existed because of publication issues, it was complicated by consortium agreements, and various issues around intellectual property. Now, what we learned from the ToxBank project, we've transferred to other projects. The succeeding EU ToxRIS project, which is one of the largest alternative projects in Europe, the legal barriers have been simplified. There's a program-wide data management plan. More attention is being given earlier to the information context and requirements of industry and regulators at the start of the program, as captured in case studies to drive this applied research. Nevertheless, there remain numerous hurdles to enabling organizing the data input into a sharing infrastructure while maintaining standards and harmonization. Tool development helps in part, but cultural developments are also needed in terms of better inclusion and higher value and appreciation of embedding data science into this applied scientific research. We still need to tear down the walls between experiment and data analysis and encourage faster knowledge flow between groups to accelerate innovation success. We also need to integrate in silico and in vitro data on safety evidence that is accumulating on case studies and associated with knowledge constructs as created by the adverse outcome pathways that are being developed within the OECD and other groups. We need to provide feedback to subsequent research iterations on the knowledge gaps and the data filling to reduce the uncertainty that we see in our experiments. And we need to continuously improve toxicokinetics and toxicodynamic models so we can support a stronger extrapolation for risk assessment purposes. The work to prepare high-quality data sets and related metadata has a clear return on investment downstream in data integration and knowledge analysis and risk assessment when we go to understand the data in terms of its perturbations on biology. But we need to reward and motivate the upstream effort. So OpenTox was initially a greenfield project. We had an empty whiteboard. In its second generation successor, OpenRiskNet, we're moving the community forward, driven by case studies to develop an open engineering infrastructure supporting many aspects of risk assessment. We're capitalizing on our learning from the early years. In addition to being interoperable and extensible to data, the project is also extensible to people through an associate partnering program. We've been able to build an interoperability layer, providing harmonized access to different sources of data, including European projects, data from American agencies, and even databases in Korea and Japan. Our progress is driven by the development of best practices. Openness enables innovation. Transparency is needed to drive acceptance of these new methods, and interoperability is needed to connect people and data. Attention to detail on the description of data, its processing and analysis, across both experimental and computational science, will provide a foundation for the reproducible science framework and solutions that we have been lacking. Now, within ToxHQ, we've taken an agile approach to application development, driven by collaboration with industry and a strong understanding of their knowledge and needs. For example, our integrated testing strategy for skin sensitization is based on a combination of modeling and in vitro assays organized against a knowledge construct, the adverse outcome pathway for skin sensitization. Against this, we can form a knowledge-based assessment, weighing the evidence with Bayesian network statistics, which quantifies uncertainty and gives a confidence to the assessor using the information. Important also, as shown here, is although these scientific methods are available and complex and actually quite difficult to reproduce, we have been able to design an application that is user-friendly for the large number of users who need to use such a tool in a practical sense and may not have the time to understand all the methods used in it. Crackit is providing an industry sponsor-driven framework to bring teams together to work on solving problems involving new products and better business processes with 3 R's impact. Such open innovation initiatives promise advances, but lack of access to data can impede the progress on such programs. Our discussions at the 2017 Global Summit in Regulatory Science in Brasilia made it clear that reproducibility is a major issue in the adoption of new methods in regulatory science. Examples include the use of validated predictive models, integrated testing where in silico and in vitro data need to be combined in a weight of evidence, results generated from complex computer simulations, analysis of an increasing amount of genomics data, and meta-analysis across heterogeneous data and multiple studies. So evidence from these methods can be used to support decision-making on efficacy, simulations, and outcomes, but the question remains, what are the best in silico practices for regulatory acceptance and use of such data? One such practice OpenTox is supporting within the EBTC Tox21 project is the construction of evidence-based toxicology workflows supported by knowledge interactions from heterogeneous sources of data, including in vivo and in vitro data, human adverse outcome event reporting, and systematic review of the literature. Biology has inherent variability. Running an experiment in triplicate may not be reproducible. Results that depend on cell lines and even different batches of cell lines. Documenting the processing of the data and workflows with computational protocols and socializing the data reliably with knowledge constructs from systems biology provides a basis for an emerging knowledge framework of systems toxicology where we try to make sense of this biology. Container technology is enabling us to take resources such as algorithm pipelines for such bioinformatics analysis and send the processing capability to the data sets, thus overcoming current significant barriers to data access, such as those posed by privacy and ethics concerns. The in silico toxicology protocol consortium is taking the learning from the initial acceptance within ICHM7 of an in silico protocol for regulatory purpose that has been accepted and is generalizing it to the accelerated acceptance of further protocols. Care for our environment is deeply rooted in our traditional values, combined with modern science and technology to ensure a better environmental care science, but also an environmental care industry. So at businesses like at Douglas Connect and other like-minded organizations, we see the promise in translating such science into a safer world by design. I'll give some insight into this area of nanotechnology, where the research project called eNanoMapper has taken an OpenTox approach to build an infrastructure to assess the safety assessment for nanotechnology. It's created a workflow for ontology integration that reuses vocabulary from different disciplines, such as chemistry and biology, in addition to adding new ontology to describe nanobio interactions. The effort is being further developed as a community infrastructure within NanoCommons, it just started this year, to extend the ontology work to describe fate and exposure concepts and related terms. NanoCommons is an example of a new OpenTox-like initiative where we attempt to bring many initiatives together from around the world to share knowledge and evidence on nanotechnology and its safety assessment and use, an area of great economic value. By providing access to harmonized protocols, data, and models, by being fair, we can integrate data and knowledge into relevant case studies, which can support decision-making, for example, in crisis situations, as we simulated at this EU-US workshop in Arlington in 2016. Looking forward, I see our challenges and activities putting many diverse resources on the same table. Collaboration and community will support conversations and knowledge activities leading to strong and rewarding outcomes. And so I see a promising road ahead where gathering resources and forces together, we'll have a better chance at designing both new products and at the same time having a safer world. Thank you.